I hope you've had a very productive week. I think I know this afternoon is going to be very exciting for you. We're so pleased to welcome Dr. Kevin Stone to serve as the speaker for our second Presidential Leadership Colloquium. You know, our goal for this colloquium is to bring some of the top people from around the world that are leading in their professions and bring them here to our campus and to explore what the opportunities are for the future and to open all of our eyes. Dr. Stone is a physician, an orthopedic surgeon, a clinician, and very importantly, a researcher. He's lectured and is recognized internationally as an authority on cartilage and meniscal growth, replacement, and repair. He founded the Stone Clinic, and his busy clinic treats people from around the world who come for biologic joint and tissue repair and robotic artificial knee replacement. He also created a nonprofit foundation to support research called Stone Research Foundation in basic science and outcome studies that are focused on cures for arthritis and techniques to speed the return of athletes to sports. He served for the U.S. ski team, the U.S. Pro Ski Tour, the U.S. Olympic Festival, and the U.S. Olympic Training Center in his spare time. He was born in Providence, Rhode Island, graduated with honors from Harvard College, went to University of North Carolina to get his MD from the School of Medicine there, and has served in various internships and experiences at Harvard and Stanford and other great places before going into full-time practice. We had the opportunity to visit with him and his research associate, Shadera Sadler, who's here today, last night, and I know many of our faculty have had opportunity to visit with him throughout the day. It's a pretty amazing story, so Doctor, welcome aboard.
We're getting set up there. So first of all, I want to thank Dr. Guys, Dr. Dua, Tom O'Neill, phenomenal generosity and warm welcome. And it's an unbelievable opportunity for me because I've interacted with people across disciplines who are open-minded and interested in collaborating. And you've done a brilliant management technique or style of somehow leveling the egos of most universities and getting people to see each other as peers. Oops, there we go. We're active now. It's so unusual, and it's very much appreciated. And it's the kind of thing that's necessary to change the typical paradigm of how do universities move forward, how they attract more resources, how they do things differently than we may have done in the past. And one of the things we discussed in one of the meetings today was, how do you open a huge funnel here? So one of the jokes about those of us who've trained for enough years in Cambridge was that a lot of great ideas came out of Cambridge, but none ever went in. And that's exactly right. And the difference of what you've done here, and therefore what the potential is to open up your tremendous resources and collaborative attitude to say, hey, look guys, do your work here. Whether, as someone who's started companies, I've taken venture dollars, equity dollars, and poured them into early stage research programs and CROs that were incredibly overpriced and siloed, where had I thought to bring them, or had the ability even to bring them to a place like this, where I could pitch the idea, explain it, the funding is the venture funding anyhow, but you bring to the table students, faculty, people of all interests who can look at that idea and say, hey, look, here's how to make it better, or here's how to expand it, or sure, we can do that veterinary work here, or that lab work, or that computer science work, and we'd love to do it because we'll get students involved, we'll get faculty involved. So opening, sort of inverting the normal program, rather than going out and looking for funding for research for ideas that are already here. Think about opening up to having funding come in and people and ideas come in because you've created a platform in which people can interact in a very lovely way. And I think if you build on what you've so brilliantly done, all of us who start companies will say, hey, I want to do the preclinical work there, and then that will grow in another way too. So it's been a great pleasure to see that. And so what I'm going to do now is I'm going to share a lot of work that we've done with you. I'm going to turn this way so I can see you. And I'm going to race through a fair amount of work just because, again, my whole approach in coming here was expose you to a wide range of things we've done, because if I get one person in the audience to say, I know how to do that better, or I know how to help you do that better, or provoke an idea, then the whole trip's worth it. And so I'm going to go through a lot of things, but at the end we'll leave enough time to ask any kind of question. And so I framed it from my friend Tom O'Neill there, how to have him drop dead at 100 on his horse. And that sort of identifies how we approach patients. How do we see a patient and say, hey, look, you've got an injury. Let's use your injury as an opportunity to help you be fitter, faster, stronger than you've ever been. And if I can do that well, which means interfering on both nutrition, mindfulness, fitness, helping repair their joint, getting them back involved in an exercise program, then we can partner with every patient that I see as a patient for life, which is the idea that we like to generate. How do we help you not just fix the problem you have today, but how do we help you get fitter and better for your entire lifetime? So let's go through that. So who am I? I'm an orthopedic surgeon, scientist. I focus on keeping people active, rebuilding joints biologically and bionically, and I'll explain that as we go along, in ways that permit sports. So we want people to be active, and your sport might be just shopping at the mall, but at least you want to be able to walk and go there. So sport can be defined as you want to define it, with a goal of having you die healthy, falling off your horse at 100. I have no commercial conflicts in anything that we're presenting in this talk. So when you get hurt today, I can either help you get relieved of your pain, but you may then develop arthritis later on in life. 50% of people who have an ACL rupture develop arthritis. So think about your college kids. 50% of the kids here who tear their ACLs will develop arthritis. Or you can get restored, or you can be made better than you've been in years. So which do you want, right? So welcome to what I call the anabolic era of orthopedics. I want to encourage you to see yourself as an athlete in training, not a patient rehab. And to do that, I need to understand who you are. What are your goals? What do you want to do? I need to use tissues and devices that make you better than you've been before and help you re-enter the world fitter, faster, and stronger.
We are all aware of the risk of cancer, but there's another disease that's destined to affect even more of us, arthritis. Cancer may kill you, but when you look at the numbers, arthritis ruins more lives. Assuming you live a long life, there's a 50% chance you'll develop arthritis. And it's not just aging that causes arthritis. Common injuries can lead to decades of pain until our joints quite literally grind to a halt. Desperate for a solution, we've turned to engineering to design artificial components to replace our worn-out body parts. But in the midst of the modern buzz around the promises of a bionic body, shouldn't we stop and ask if there's a better, more natural way? Let's consider an alternative path. What if all the replacements our bodies need already exist in nature, or within our own stem cells? This is the field of biologic replacements, where we replace worn-out parts with new natural ones. For instance, in your worn-out knee, we create new cartilage using a paste graft of your own stem cells and replace the meniscus and ligaments with ones from a donor. We are now moving into super biologic enhancements, where we add novel cells and growth factors to the biologic replacement tissues. And our options extend beyond human tissue. By treating animal tissue and seamlessly integrating the best elements of the animal kingdom into our human anatomy, we create super biologic replacement solutions and merge the best of evolution from different species.
So that's one part of our goal. And so what are my tools for helping you do that? So that my platforms are non-operative, physical therapy, lubrication, stem cell-derived cells, which we're going to talk a lot about, and growth factors. Biologic replacement, replacing the tissues, or robotically using implants to replace the most worn out knees. And of course, having research be the background of everything we do. I fundamentally don't believe that a doctor should do anything to you or put anything in you that they don't know how it works, or at least be asking the question to figure out why it works and what's the outcome of that study. So that's why the research infuses everything we do, and hopefully you're infecting your students as well, that research in medicine and research in much of the programs that they're going to go into in life are intimately intertwined. So from fitness and physical therapy, every patient coming to our clinic sees our fitness team and physical therapy team. It's an essential part of recovery. They see themselves an athlete in training and infusing that idea of becoming better. And essentially, I'm a drug pusher, as I like to say in a colloquial way. I'm trying to push endorphins, testosterone, adrenaline, pheromones, all the things you get from exercise, because that's what makes you healthy, keeps you healthy, solves that ACL depression syndrome that after you injure your knee and 3 weeks later, you're just kind of sick of the whole thing and you want to be back to your own life. So the tools that we have to lubricate your joints have been hyaluronic acid, the natural lubricant of the joint. And we've been using that for a number of years now. And we observed that 30% of patients would have a super response. Just a hyaluronic acid injection, they'd be great. 30% sort of half and half, and 30% didn't respond. And we clearly wanted to do better than that. So our choices for stimulating your body's own cells to produce more lubrication in the joint and to shut down inflammation are fat, because your fat's highly vascular, PRP, which is where we take your blood and spin the blood, grab the platelets, and release the growth factors from the platelets, BMAC, which is where a surgeon puts a needle into your bone, draws out the marrow fluid there, spins it down. Or, most exciting lately, has been amniotic fluid. I'm going to explain quite a bit about that coming up here. We very rarely, if ever anymore, inject cortisone. Cortisone shuts down cell metabolism, weakens the tissue, accelerates the arthritis. So in our anabolic year of orthopedics, we want to stimulate you to heal. We don't want to shut down the healing and accelerate the arthritis. So when we do tissue replacement, so if you tear your ACL, it's a bummer that our athletes take a year to come back from their ACL injury. So what can we do to accelerate the healing, the remodeling of that tissue that we bring back? We can preload the tissues with anabolic factors to accelerate that healing response. We're just trying to wrap some data around that right now to see how much that works. So PRP, as I mentioned, the platelets from your blood, we can take them, spin them, inject them. They have chemotactic factors that recruit your body's own stem cells to the site of injury. Your body's own stem-derived cells. And let me clarify the word stem cell for just a minute. So unfortunately, we and everybody else use the word stem cell as the cell that we were injecting to induce tissue regeneration into your injured joint or injured tissues. We now know that they're really not stem cells. They don't regenerate that tissue. What they do, the cells that we're taking off the vessels, is that they are medicinal cells. They drive the healing response. And so it's important to understand it because you have millions or billions of these cells throughout your body, and let me show you that in a second. So we're not truly injecting stem cells anymore, and in fact, we're not even injecting cells anymore. We're now injecting chemotactic factors to draw your body's own repair tissue, your own repair cells, to the site of injury. So if I'm going to give a lubrication injection, I always give it with an anabolic factor. So stem-derived cells, they're not true stem cells. They're cells which we call MSCs. They used to be called mesenchymal stem cells. They're now called medicinal cells, directing healing, and they express these proteins to shrink inflammation and to stimulate new cell growth. And they're recruited to the site of injury by these factors that we inject. So these cells live on vessels all around your body. And the reason why people took fat was because fat is very vascular, so it has tons of vessels, so it has tons of these cells living on the walls of those vessels. They're not stem cells, but they're the next derivative down, and they direct the healing in your body. And so the research in this anabolic area is we want to understand the impact of these cells and growth factors. And we're currently running an IRB-approved trial where what happens when you come to the clinic, I aspirate fluid off your joint, run profiles in that fluid, and then give you one of four things, and I'll show that to you in just a second. So the study is that we want to know hypothesis 1, did the bioactive factors increase the lubrication in your joint? And number 2, do they shift the joint chemistry to a more anabolic, less inflammatory state? And the way we want to answer that question is we first said, what's in normal joint fluids? These are the factors that live in all of your normal joint fluid. And some of these factors are anabolic factors. They stimulate tissue healing, and others shut down tissue inflammation. And in arthritis, there's an imbalance in these factors, or a disconnect, so you have more of a degradative environment and more of an inflammatory environment. And so we can look at these anabolic factors, some of which are much more potent at drawing your body's own stem cells, and we can look at these degradative factors, some of which also draw the body's own stem cells when you have a site of injury. And so if I'm going to give you an injection into your joint, I want to give you an injection of the most potent of these factors that recruit your body's repair tissue and shut down your inflammation. So what's the most potent source of growth factors in anyone's body? Well, it's amniotic fluid, because there's nothing growing faster than the fetus. And the reason that the mother doesn't reject the fetus, even though the fetus is only half the mom, is because the cells and the growth fluid and the growth factors in that amniotic fluid are potently immunomodulatory. They shut down inflammation. They shut down the rejection response. So healthy women coming to a C-section donate their amniotic fluid just before delivery of the baby. That amniotic fluid goes to the tissue lab and then is processed in several different ways, and the processing affects whether or not we get cells or we get just growth factors. And so in this amniotic fluid, we have potent immunoregulatory and anti-inflammatory factors. And the functions of the amniotic fluid are protection, mechanical support, nutrients, anti-inflammatory, anti-scarring, antimicrobial, everything you could ever want in a fluid to be injected into your painful joint or tissue. And the amniotic fluid has 2 to 50 times the concentration of the factors that we want than does your own blood platelets or your bone marrow or your fat. So it's the most potent source of an anabolic injection that we can give. And in the lab, we try to grow these cells and see whether or not it's the cells that are doing it or whether it's just the growth factors that are doing the effect. And it turns out that initially we thought we could grow stem-like cells from the amniotic fluid. However, it turns out, and I was quite excited about that, however, the more studies we did with it, the more non-reproducible it was, where sometimes the cells would grow and sometimes they wouldn't. And so we came to realize that even when we filtered the fluid and got rid of all the cells, the potent factor was really the growth factors recruiting the body's own stem cells rather than the cells that we might be injecting. So that study is just completing now, and I'll show you a little bit of that data where the patient on day 1 would either get saline, get hyaluronic acid alone, get amniotic fluid, or would get hyaluronic acid plus amniotic fluid. And then 1 week later, we would go and aspirate the joint and look to see what we had done. And then we follow the patients over time with outcome studies. The average age of the patients, these are all older patients with arthritis. And we looked at which factors were increased after we injected an anabolic. And hyaluronic acid appears to be increased after we inject this anabolic. So that may explain why do these patients get joint relief. And the factors that when we inject hyaluronic acid plus amniotic fluid, we get a bigger bump in the factors that we're interested in and a bigger bump in the hyaluronic acid. And when we inject saline, we get a decrease in the amount of hyaluronic acid. So we're starting to collect data around when we inject these factors, what happens to the joint. However, the faulty thinking in my first study here was that I could look at 1 week, when in fact we need to look at 1 month, because it takes 2 to 4 weeks for the body's own reparative cells to be recruited to the site of injury. Another slide just looking at these biomarkers that are increased. Now, what's curious is if you come to our clinic with an arthritic knee and I give you an anabolic injection and the stuff goes in the joint one day, why do you get better over time? And it turns out if I follow you over time, your pain scores improve at 1 month, but they improve at 3 months, and they keep improving at 6 months. And your COOS, which is an activity score, improves at 1 month, and it keeps improving. And your symptoms improve at 1 month and at 3 months, and they keep improving. Why does a single injection cause a prolonged effect over time? And this is the question we're trying to answer by doing our next study with 100 patients where we do the aspiration on day 1 and we do the repeat aspiration on 1 month to see is this long-term effect a result of drawing your body's own reparative tissue, reparative cells to the site of injury. So we're just pending funding to do that larger study. Pain relief, again, 1 month, improving at 3 months. So how do we explain that? So our impression was that at 1 week, these biochemical markers would be making a difference. That impression was wrong. It takes longer than 1 week. It takes a month. And that the peak derived cell time is 2 to 4 weeks. So the next study is looking at 100 patients over time, and that study is what's pending funding. So our belief is that the patient's own reparative cells are the best source of treating your body, not injecting someone else's cells or injecting stem cells directly. Let's look at operative treatment. So if you come in and say, hey doc, I lost my meniscus cartilage and now I've developed arthritis, let's look at what our options are for treating your knee. So first, we can either replace it with biologic tissue, which that's a meniscus tissue. I'm going to show some slides here, and that's what a meniscus looks like when we're putting it back in someone's knee, or we can replace it with bionics. And let me explain the pros and cons to each of these. So if you limp, you clearly can't be better than you were before. So we need to solve your limping problem. We need to solve the problem of arthritis or cartilage loss in your knee. And we can do that depending on the injury that you have, either by replacing the meniscus, regrowing the articular cartilage, or replacing the ligaments, or doing all three, called a biologic joint replacement. So we now have 2 decades of this bio-knee research where we combine these procedures together, and I'm going to show you a little bit more about that. So the meniscus is that key shock absorber in your knee. It's torn 1.5 million times in the US, and unfortunately 90% of these meniscus tears, the surgeon goes in and takes the tissue away. And by taking that tissue away, they eventually develop arthritis. And so loss of the meniscus leads to this painful wear and tear of the articular cartilage and arthritis. And the data is that 60% of the patients who lost their meniscus progressed to arthritis, but only 20% progressed to arthritis if the surgeon would repair the tissue. So we need to teach surgeons how to repair and to repair. And there's also, whether repair, regenerate, or replace, if we can get people to do that early, as soon as the athlete or the patient injures their tissue, rather than waiting until they develop arthritis, then we can potentially solve some of the arthritis problem. So again, the meniscus is the key shock absorber if you tear it, repair it, or replace it. So early on in personal history, so I'm out for a run with my mentor, and this is 1984. He looks at my bow legs, and I'd lost my meniscus playing soccer in college, and he says, Kevin, you know, if you could ever figure out how to replace the meniscus, you'd make a big contribution to orthopedics. And I said, great, I'll do it, you pay for it. So I started my lessons in funding. So my first effort at replacing the meniscus was to try to regrow it, to develop a regeneration template. And so I took cow Achilles tendon, blended it in a blender, and formed it into a collagen scaffold, working with a collagen chemist in New Jersey that I identified with after I'd spent 2 years in the library reading the world's literature on meniscus and biomechanics and how it should work, and putting quarters and nickels in the Xerox machine back then, which is how we did research. And so once I understood that biomechanics, we built that collagen scaffold. We now infuse tissues and cells and growth factors into it and insert it back into the meniscus. And so here's how the collagen scaffold gets sewn into place. So again, the patient comes in, they've torn the meniscus. The surgeon looks in the knee and says, I can't repair this, it's too beaten up. The surgeon goes and shaves it away, says, come back to me when you develop arthritis. No, we want them to say, if you can't repair it, regenerate it, try to regrow it. And there's a collagen scaffold that you can sew into place that we developed early on to do that. It got approved in the US, approved around the world. The good part to it is that it worked pretty well. The bad part, and you can see regrowth into the collagen scaffold of a normal meniscus. The bad part to it is that it's really not durable enough, and many surgeons got frustrated when they put it in. So we have a program now to see if we can build a stronger, better, more durable collagen scaffold. So if you can't regrow it, if the cartilage and meniscus is too damaged to regrow or to repair, we want you to replace it. And the replacement source today is human meniscus allograft from usually kids who fall off their motorcycles, which is why we call them donor cycles. And they donate their meniscus tissue to us to put back in someone's knee. Now, I don't plan to gross you out immediately after your lunch with surgical pictures, but here's how I put that meniscus back inside someone's knee. It's a lateral one, and this is a medial one. I'll show you. And we demonstrated that we can have live cells within these tissues, and we can mobilize cells into those tissues with various growth factors. And then here's a quick little video of how I actually do it. So here's the tissue as it comes from the bank. We mark it so I can tell the top and the bottom when we pull it in, because one of the problems with the surgical procedures is it can get twisted. Here I pull it into someone's knee through a little hole, pull it right back into the joint, and then sew it in place. And so now we've restored their meniscus shock absorber. And we want surgeons to do that all around the world. So I run around the world teaching surgeons how to do meniscus transplant. And unfortunately, because there are not that many tissue banks, not very many of them are being done. So my typical patient, ACL reconstruction, and then a microfracture, and then another cartilage procedure, and then shows up with an arthritic knee in 2004, and we put a meniscus back in. Good part to that is it works. The bad part to that is he goes back and does that. So am I just empowering my patients to continue to hurt themselves, or am I putting them back in their life activities? That's a real debate. Some patients are an annuity. Other patients, they're just going to keep coming back. It's just the way it is. Okay, so we published a lot of research in this, and clearly, we want to do high-quality outcome studies, and publication and peer review is the metric that we use and other people use to be sure that our research is going down the right pathway. So let's look at what happens to the articular cartilage. We talked about the meniscus cartilage. What happens if you just developed an injury to your articular cartilage, or you're developing arthritis where you've damaged or lost the articular cartilage, and you're too young, you don't want to have an artificial replacement? Well, we developed a technique in 1991 called paste grafting, where we graft this damaged area.
Here we demonstrate a medial femoral condylar lesion in an athletic bicycle racer. The lesion is morselized with a chondral ball to create a bloody bed. This lesion had been present for years and had stopped him from participating in bicycle racing. He had previously undergone a medial meniscectomy, which most likely contributed to the degeneration of this medial femoral condyle. The dead areas of bone are removed with the arthroscopic awl. The lesion is shaved to remove loose pieces of cartilage. Care is taken to ensure that a bloody bed is created. Graft is harvested with a trephine from the intercondylar notch, taking care to capture a small amount of articular cartilage and underlying bone. The graft is taken out of the knee, formed into a paste, and then impacted into the lesion with the trephine. No effort is made to completely fill the lesion, as these usually hypertrophy during the healing phase. Loose pieces that fall off into the joint are removed with an arthroscopic grasper.
So there we go. So then we get a chance to look back in that same patient's knee in a follow-up where we put a new meniscus back in, and here's the cartilage surface all nicely healed over. So we know that in many cases we can get the cartilage to heal. And when we send biopsies of these out to our musculoskeletal pathologist, 1/3 of them look like normal articular cartilage, 1/3 are a mixture, and 1/3 are just fibrous tissue. But we've followed these patients out now 25 years and know that we can get survival of the cartilage paste, and I'll show you that data. So our in vitro models to say, hey, does this work in the lab as well as in people? And typical in orthopedics, we go to people first, and then we go back to try to figure out, hey, why does this work? It's a little bit of a problem with orthopedics, but that's what we do. So we look in vitro models where we put cells in these matrices, and then we're able to extract the cells from the matrix and demonstrate that we, by smashing that cell and that graft into a paste, we can upregulate and produce more collagen inside the joint. And most recently, we just completed a study comparing what happens if you take an intact cartilage plug versus our smashed up paste. Because in orthopedics, we were all taught that if you hit cartilage, you kill it. So in our upside down thinking, the way we like to think, well, maybe that's really not true. Maybe you kill it later, but maybe it doesn't die. Maybe what happens, and the reason why paste grafting works, is that initially the cartilage tries to repair itself. It creates this huge reparative response. And that's in fact exactly what happens, but we've now only proven it now 20 years after we've been doing paste grafting. So we went ahead and did this little study in the lab with a team in New York where we hypothesized what's the difference between the plugs versus the paste. And we can show that if we take donor plugs versus paste plugs and smash them and then look at them over time at 24 hours and 48 hours, we've dramatically upregulated the cell production from the cells and matrix in that particular cartilage paste graft. And the genes start producing all these healing factors, and that's why the paste graft probably works. And then when we look at the histology of these, we can see normal chondrocytic production in these paste grafts. So we want to know what can we add to those paste grafts now to make them work even better, and that's another research project that we're starting to think through. So our hypothesis that smashing paste upregulates and institutes a regenerative process has been held up by what we've done. Here's an example of a patient where they have an isolated chondritic defect. We create a big bloody response. We pack the paste in. We come back and do a biopsy here at 7 months. And you can see a beautiful cartilage regenerative surface on top of the underlying bone. Here, 14 years post-op, he is still doing extremely well. In another patient, here, when we make those little microfracture holes, here you can see the cells pouring out of the bone matrix, trying to create this regenerative healing response on top. And so we're using the marrow's own cells and healing ability to pour out into the joint for healing. And even in very arthritic joints, patients who come in who's only 42 years old says, hey, I don't want to have a partial joint replacement yet. Isn't there something you can put in my knee just to help me buy time? Well, if we fracture this severely arthritic surface and look back 7 months later, we can see a nice healing response over that arthritic surface. It's not by any means normal cartilage, but pretty good enough to provide pain relief. It's a mixture of mostly fibrous tissue. But here I look back at her at the 20-year follow-up. It's the beauty of having gray hair as your patients eventually come back. And here at the time of her partial knee replacement, we can see her healed response. The rest of the joint has gone to hell, but the healing response has stayed pretty well. And so even in kids with OCD lesions, these giant lesions that they sometimes get, surgeons sometimes try to repair them with screws and other techniques, and when it fails, they show up in my office with this big hole in their knee. And we go ahead and paste graft that big hole, and we can stimulate healing in that huge hole, as you can see here. And the biopsy shows that it's healed reasonably well, but not with normal cartilage. But here in his 10-year follow-up, he has no pain and an activity score of 5 on a 0 to 10 scale. And in a 25-year-old here, the huge defect, that OCD defect, the day I saw him back in my office, he had run 28 miles in 2 days prior to his follow-up. So we know we can get these to heal pretty well, even if not perfect. When we look at our articular cartilage outcomes over 10 to 23-year follow-up, and again, this is the importance of always being able to ask the question of anything you're doing to a patient, does it work? So following these patients over a lifetime, and we look at the fall-off over time, but we know we can get a 17-year benefit from these articular cartilage paste grafts in a population average age of 49 with significant arthritis in their knee. And so we published a sequence of these in different kinds of presentations that the patient arrives with, whether arthritis or isolated defects. And so when we can look at our combined, what happens when we put a meniscus and articular cartilage back into people's knees, in the first 343 of these patients, following them over time, we get an average benefit of about 17 years with a significant improvement in patient outcomes. So we're pretty good, but we're not perfect yet, and we need to be a lot better than that if we can be. What happens when they tear their ACLs, as many of, unfortunately, the students here will do in sport, many surgeons still take their patellar tendon or their hamstrings. We don't believe you should have a second site surgery in the 21st century, that you should be robbing one part of a patient's body to rebuild another. We need to be better than that. We're quite biased about using donor tissue, because when I looked at my first 15 years of bone, patellar tendon bone, the patient's own tissue, and compared them to my next 15 years of just using donor bone, patellar tendon bone, there was no difference in the outcomes. We no longer use the patient's own tissue to rebuild their ACL. We also then started a program, hey, could we use pig tissue? Pigs are young, healthy, and strong. What's the problem with pig tissue? It's obviously immunology. We built a program around stripping pig tissue of the antigens so that we could use them in people. We took it through primate studies, then a pilot study at our clinic, and then it went out to a wide clinical trial in Europe. It got approved in Europe for a CE mark approval for sale in Europe. And then what produces gray hairs is that one of our investors was disgruntled and drove the company into a bankruptcy. So it's now sitting waiting for another round of funding, and it will come back in life. But pig tissue and animal tissue as a source of replacement tissue is very promising for a host of reasons. But right now, we're just using human tissue here. So human tissue, an ACL, you're a young athlete, ruptures it. Surgeons sometimes use Achilles tendon or hamstrings or patellar tendon where it's bone, patellar tendon bone. We believe you should only use this as donor tissue. We don't take the patient's own tissue. However, every ACL tear, when can the athlete be back? Why does it take a year for the athlete to come back from an ACL? Can we accelerate the rate of healing in this avascular graft? And we believe that the solution may be, and we don't have data yet to say, would be to preload these grafts with these recruitment factors to draw the body's own reparative cells right into the graft. And in my fantasy, and this will be great for the technology team here, in my fantasy, and this is only a fantasy so far, we make this little machine that you give to the surgeon, and pre-programmed into this machine is the exact amount of time, pressure, and temperature that you should apply to that specific tissue in order to load it with just the right amount of growth factors in order to then put it in the patient's knee. So a little bit of work for us all to do to get to my ideal machine, but this is where I think we're going. You'll never put tissue back in a patient that you don't optimize in order to accelerate that healing. And that's the work that we're building towards that, as you can hear in each of these tissues. And we know that we can drive cells into them because we do these tests where we look at dead here and live cell are green. I know I can drive cells and growth factors into tissue. The question is how much and which factors should I use? So again, just looking at my fantasy machine here for grafts or meniscus or ligaments or tissue matrices, or if you're having your shoulder rebuilt with tissue, any one of these things I want to make that repair process better than it is today. Okay, so a typical patient of mine, microfracture, which is a repair tissue, has a PCL injury and then an ACL injury, and then for her other knee does her ACL repair and reconstruction of her ACL and allograft, and then a meniscus. And of course, she goes back and wins X Games gold medal about 8 months after I did that first sequence of surgeries for her. But again, as I said, some patients are annuities. You know that every once in a while they're not going to land that thing perfectly. But there's nothing I could ever say to that patient that would prevent her from doing that. So let's get off biology for a minute and look at bionics, because the promise in bionics is that you would be much better than you were, and we've heard that promise now for several decades, that you're going to have a stronger skeleton, you're going to have better hearing, you're going to see like Superman would see. And so what's the status? Well, in orthopedics, we're very crude. We used to use saws and guides, and now we can use a robotic arm. So I can do a CT scan of the patient, build a model of their knee before I ever touch their knee, plan to where these implants should go inside their knee, and then at surgery make a much smaller incision, just use a robotically guided arm to burr away only where the implant should fit on the femur and the tibia, not touch anything else, not touch their ligaments, not touch any other tissue, and then put the implant exactly where we planned it on the 3D model on the computer. And this is just way more accurate than I could ever be. So I did these partial knee replacements for up until about 8 years ago in my clinic, and I thought they were pretty good, but once I got a robot, I realized how far off I was in many of those cases, and the robot is just so much more accurate. So we plan these things, and we do it for the inside of the knee, the outside of the knee, or just the patellofemoral joint, or sometimes combinations. It's called a partial knee replacement using a Mako robot, and they're my most happy patients because they're usually in the 40 to 70-year-old age group. They want to ski and climb and run marathons and do all kinds of things. 80% of the patients who have been told that they need a total knee replacement do not. That's not my data, that's data from England. Most people who have been told they need a total knee replacement have only worn out part of their knee. And so the question is, can that part be rebuilt biologically, or can it be replaced in parts rather than doing the entire knee? And a partial replacement preserves the ligaments, makes the knee feel so much better. So whenever we can, we'd rather do a partial. We'd rather do the least amount of surgery possible to get the patient the most athletic performance and pain relief. Now, this particular guy went and did a huge bike ride after his partial knee replacement and all kinds of other crazy stuff. Because my patients seek me out in order to do this stuff, and so there's no way we're going to hold them back. So this is an example of another guy who comes in and say, hey, I'm a senior racer on my bike. I want to keep racing hard. I can't miss this season. Can you fix it so I can be back on my bike in a few weeks? And where most mortals wouldn't, he would. In patients, however, when the joint disease is too great, and they really are destroyed in all the compartments, what's changed in knee replacement is now with these robotic guidance into these severely arthritic knees. Got a little water? We can put these implants in now so accurately that we don't need to use cement. One of the problems in the past was that when we put the implants in, you needed to cement them to the bone. And so surgeons would tell patients, don't run on it, don't climb on it, just preserve your knee, and yes, you don't want to do a second one. Well, the problem in telling patients to not run and not be active is that, especially in women, they become more osteoporotic over time and they lose muscle mass. And so we tell patients now exactly the opposite. We want you to increase your exercise, increase your skiing, increase your climbing, because the more active you are, the more you'll build your bone, the less likely it is to come loose, and the more you'll enjoy your knee. By not using cement, I never have to worry about you knocking the cement loose. The other thing that surgeons told patients is, don't go too hard on your knee, you're going to wear out the plastic. So every time I gave a lecture at a knee arthroscopy meeting, I would ask the audience of surgeons, how many of you have ever seen a joint replacement worn out from sports? No one would raise their hand. But wait a minute, you've told your patients for the last 20 years don't play sports on their joint replacement. So why? And the reality is they don't wear out from sports. They wear out from either osteoporosis, muscle weakening, or the cement coming loose. So I think we can solve those problems now. So new versions of joint replacement with a robot eliminates that cement and permits people to go back and do sports. So let's look at other items. So the path forward from where we are today to where I'd like to go to is, of course, repair, regenerate, replace tissues as soon as they're injured. Drink water whenever you can. See what we can do to arrest arthritis. Repeat the regenerative therapies. We don't know how often should you come back in to have a joint injection for your sore knee or joint. Harness self-therapy, meaning your body has billions of these stem cell-derived reparative cells. We want to harness that repair process. Prevent, treat, and eventually cure arthritis. That's our mission. So your tissues are still 70, even though you maybe feel like 50. We want to take the strongest tissues possible. So right now it's human allograft. Eventually it'll be more pig tissues. If they're metal and plastic, we want to use those that will grow into bone. Okay, so that's what we've done. Now what are we doing in terms of research going forward? So our next set of studies that we're very excited about and here talking about, hoping to find people here who might have an interest in one way or another, are first of all, if I hit you with an axe, otherwise known as surgery, within 8 hours you start developing significant muscle atrophy. And it's the #1 thing that people complain about for months after a surgery or an injury. They've gotten their joint repaired, but they've got this atrophied leg. And they have to go to lots of physical therapy and training and fitness and loss of work. So I want to see if we can prevent that atrophy. When I hit you with an axe or surgery, there's an enormous cortisol release from your brain, stress release. That cortisol binds to muscle receptors, and we think that that's what instigates the muscle atrophy. So the first study we have proposed is we want to block those muscle receptors before I take you to surgery. And we'll give you a second injection of this 3 to 4 weeks later. and see whether or not we can block that atrophy. And if we can block it even by 25%, it would be a big contribution to people. The second study we want to do is an equine study looking at articular cartilage paste grafting. Equine because it's a large animal, and the large animal, the horse model, is the best model for cartilage repair. And we want to show in animal models that paste grafting works so well. We've shown it in people, but it's not seemingly good enough to get enough surgeons to be using it around the world. We want to do a human clinical, a wide clinical trial of paste grafting. We're just starting to get that going in Poland, we believe, next year. We want to expand our growth factor trial, the one that I showed you early on, to a 100-patient trial with an aspiration of the joint at 1 month, not at 1 week, and see eventually we're going to know exactly which factors should I give you, the patient, what's unique to you in your joint fluid, and what are you going to respond to. And we think that we can start getting to that answer. What happens when I inject HA or inject a growth factor in your knee? What happens to your knee biochemistry? And so that's that 100-patient trial. We want to do an infectious disease trial. And let me explain that for a minute. In some people's mouths, there's an organism called Porphyromonas gingivalis. It's a bacteria that hides in the periodontal tissue. And there have been some reports that it may be associated with Alzheimer's and rheumatoid factor in arthritis. What we want to know is associated with some osteoarthritis as well. So people always said, listen, I got osteoarthritis because my mother had it, and my father had it, lived in my grandfather had it, and it's a genetic disease and I got it. Well, if it were a genetic disease, it would have been factored out by now, because there's no advantage when the tiger is chasing after you to have an arthritic knee. You'd be gone. So I think that you might have gotten arthritis because your mother had it and your father had it because you lived in the same microbiome environment as your mom and your dad and your grandparents. And so some percentage of arthritis, just the way some ulcers now are treated with an antibiotic, some percentage of osteoarthritis may be infectious, and that particular organism is very hard to culture, and we're looking at 11 different hard to culture organisms by taking tissue from people's knees who are coming for joint replacement and doing PCR testing looking for these hard to find organisms. And if we find them, and if they're treatable, then we'll find that percentage of people with osteoarthritis who may be cured with an antibiotic. We want to make a more durable collagen scaffold, because I showed you the collagen scaffold that I made back in the late 1980s for meniscus regeneration, and it was accepted and sold in the US, but it's not good enough, which is why it's not used widely. We need to make a more durable collagen. We're working with a group at Northeastern on a liquid crystal collagen, which makes it more dense, and maybe that will be a solution, but there may be folks here who want to work on that as well. We also have a lab and a chip collaboration. So today I was exposed to the team here that's working with labs on the chip, and we want to take the articular cartilage from your knee or your tissue and run studies. What do you respond on that particular cell in your body to which growth factor? And we can do that on a chip now rather than doing it in an animal or in a person. So quickly going through those, so here's the muscle atrophy. It starts within 8 hours. We want to inject testosterone, see if we can block that. Large animal model in horses for paste grafting. A wide clinical human trial as well. An injection study we want to expand to 100 patients at 1 month. An infectious disease trial, how much of OA is infectious, and that would be it's a 200-patient trial that we're just starting now. A liquid crystal, a lab on a chip model, because each of these cells we should be able to test against on a chip these days rather than in an animal model. So I can take your synovium or your articular cartilage or wherever at the time of surgery, put those cells on a chip and test them against various drugs or various anabolics. And that's the lab on a chip model. And that's currently in a collaboration with a group in Austria, but it turns out you guys have some of the similar skill sets here too, and we should be able to do some of that work here. So if you want to live and play to 100, my advice to you is live and play now. If you get injured, repair, regenerate, replace the tissues as soon as possible. Don't sort of live with it. And kind of take your head out of the sand. Don't think, hey, I'm going to be just fine. You're not going to be fine. If you've injured your tissues that are critical to your joints are not going to work well over time. How do we get so much done? I always get asked. So I would say to students and fellows that are trying to mentor that, listen, meaning is the antidote to burnout. Always try to do something bigger than yourself. I personally find a lot of time in every second, and so that's what we do. We welcome fellows, students, visiting docs. You know, we love having people come by and try to share some of our enthusiasm and curiosity about what we're trying to do and see if we can make it better. And of course, in any situation, I have lots of collaborators, and Shadera Slater's here in the audience, who's been a huge help on all this data collection and presentation and work. We're also using a system, a link by Links Care, that may be of interest to some of you. It's a data mining AI-driven software program to look at our 30 years of patient records, which we can dump into this data lake and then do data mining from it, and eventually AI-guided patient care decision-making. Thank you. Open for questions. Yeah.
What materials are you using for your cementless implants?
So, yeah, the question is about materials for the cementless implant. So, right now, several companies make them, but Stryker Triathlon makes a bony ingrowth cementless implant that has worked well in our hands. And they'll get better, but they work well now.
The hips work about the same as your knees.
So I don't do hips, so I don't have the hip data. But what I hear from my friends is that hips really do work extremely well these days. They're not using cement anymore most of the time. They're doing anterior approaches, so they're not cutting the muscles anymore. And our hip patients seem to come back very quickly and do quite well. So I think the hip field has actually been ahead of the knee field for a while.
Materials for the implants, are they still primarily titanium, vanadium?
Yes, cobalt chrome for the femurs, titanium bases for the tibias, for the mechanics, and high molecular polyethylene for the plastic inserts. I'll tell one quick story. So last few years in skiing, and those of you who ski, you remember the PTEX bases, the bases of skis that we always used to all repair? That PTEX is high molecular polyethylene. And so a ski company came out with a wax that, with a new treatment for the base of skis that penetrated the PTEX, and so that you didn't have to use a wax and increased lubricity of the PTEX ski base. So typical me, I went to them and said, hey, if you can do that for skis, can I do this for total joints? Can I take your treatment of ski bases, apply it to the plastic material, the high molecular polyethylene material we use in a total joint, and make it more lubricious? So we started a study with a guy at Dartmouth who's the expert on the biomechanical factors of implants, and applied the treatment. And our first go-around made it worse rather than making it better. We hope to do another go-around and figure out why that happened. But just to know that, you know, look around at the places that you're active in life and apply things from outside of your field to your field, and sometimes you get lucky and they actually work, but not always.
If you responded well to fat cell injections and had a 6-month
Yes.
free, do you see the same type of response in Bionics?
Yes, because it's just a higher concentration of the same growth factors. And maybe back to the hip question, yes, we inject hips the same way we inject knees. It's a joint response, the same potent anti-inflammatory and lubrication ability. It's just no fun sticking needles in fat and in your bone when you can take the stuff right out of the freezer now. Yeah.
I know you said you leaned into the reasons why a single injection would have such a long
Yes.
effect, but what's your hypothesis?
That we recruit the body's own stem-derived cells to that site of injury, and that it kicks off a potent anti-inflammatory and reparative process and stimulates the lining of the joint to produce more HA. And we think that by looking at that at 1 month rather than 1 week, we'll be able to show that. It's the only thing I can think of that would explain why do people come in. I have so many patients who come in before ski season and say, hey doc, you know that injection you gave me last year, I can tell you the day it wore off, my knee felt great for a year, I'm going skiing, just give me another one of those lube jobs. And it's decreased my surgical practice, because those patients in the past would come in and say, hey, my knee's too sore, replace my knee, or fix it. And now they come in and say, hey, give me another one of those injections. And it's bizarre how well it works for some people, just not everybody. And so we need to figure out who's the right person and why do they respond to one thing versus another. And that's the goal of the research. Yeah.
Does any of this, have you seen this applied to arthritis from the autoimmune disease?
So I'm very biased. This is a hot topic in autoimmune disease, and I'm going to tell you something very controversial that not everybody shares, so please don't be offended if I don't believe in autoimmune disease. I think that every one of those autoimmune diseases is an infectious agent. We just have not identified the agent that kicked off the immune system to flare. Because genetically, autoimmune doesn't make any sense. Why would your body turn inward against itself? We don't see it anywhere in the animal kingdom. It just doesn't seem logical to me. Sorry. So I think that, as I mentioned, 40% in your nose, 60% in your gut code for things that we don't know what they code for. So I think they're coding for agents and organisms that kick off the immune system, and that we will get better and better at figuring out what those agents and organisms are for Alzheimer's, for arthritis, for so-called autoimmune diseases, for Crohn's, for a lot of the digestive microbiome disorder diseases. And that's my own bias. We need science to prove it, so we need all of you to help, and people are interested in that part of it, but I think the microbiome is the great source of our new information about how disease and health actually work.
So, but would the injections be
Good question.
profitable?
Yes, good question. I'm sorry I didn't answer that part of it. Probably yes, because they're potent anti-inflammatories. We just don't have very much experience doing it, but the potent anti-inflammatory component of the autoimmune, the part that bothers the patient is the inflammatory process that produces the pain, that whole cycle. So seemingly, if you could shut down some of that inflammatory process, you would help the patient. You just have to try, and the likelihood of doing harm is really low. And so, the beauty of in medicine in particular, we're all taught to do no harm. So to have this potent therapy, this potent anabolic science, where it doesn't look like we would do any harm to the patient, and yet have huge benefit is a dramatic change in our lives. Is what is such a jazz. And curiosity about why and what works and how to optimize it. Yeah.
So with your magic machine,
Yes.
your view of the materials of the cells, would the material at this time need to be uniform or have gradients, for example, gradients of porosity, or one uniform set of materials properties? Because I'm thinking chemotactic, cell biologists, we had a chance to talk. These gradients of growth factors go from low to high or high to low. We have gradients that repel cells, gradients that attract cells. And so do you think the material itself would need to have gradients, or start with a uniform property?
So that's why I asked you the question when I was in your lab, what happens when you put current across that?
Right. Well, we're trying to work on it.
Because current or magnetic field will draw your copper-based implants there to one side or another, or across a tissue. And so the answer is we have no idea whether or not we need to uniformly penetrate, which we probably don't anyhow whenever we inject things. It's probably very non-uniform. But what happens in ACLs is the body remodels an ACL reconstruction with vascular ingrowth. And by about 3 months, that ACL reconstruction is at its weakest point because it has all that vascularity in the tissue without a lot of tissue remodeling. But the athlete feels good and says, hey, coach, can I go back and play? And you're looking at him at 3 months and say, well, your knee looks great, but we know it's at its weakest point. Well, the question is, even if we're not uniform, can we penetrate that tissue with growth factors on day one or other things? And it could be even magnetically based particles that you then drive by MRI into tissue. Another thought for you. In order to sequentially augment the healing process of that tissue. And you may give an injection at 3 months to that patient, inject the factor of the week, drive that factor in by magnetic field, electric pulse, ultrasound, whatever your stimulus is. It won't be uniform. In terms of matrices, if you were making them going through an FDA process, however, the FDA would require you to be quite uniform and understand the biology. So depends on which path you're going down. From a materials standpoint, probably yes. From a tissue standpoint, probably no. I hope I've inspired some of you to think about this stuff and let you know that we're very excited to be here. We're excited for the patients we get a chance to take care of. And we're very excited for the science that we get to do. The science drives the clinical practice. And so as much as we can infuse the students that we're all lucky enough to influence, that be a scientist first and then be whatever else you want to be in life, then you'll be kind of understanding what works and what doesn't work.
Dr. Stone, thank you for your incredible and inspiring story. It's, we've been here able to take advantage of you all day and even last night, and learning from you. I know I challenge our faculty and students to take him up on his offer and show something that you think can be better. And I know he'll feel that this journey was a success if we could accomplish that. But it's been successful for us. Thank you for taking your time to be here this busy time for you. And I'm sure we've already had some local people that have traveled out to California to visit with you and to stay there for a little bit of treatment, and I'm sure we'll have more headed out that way in the near future. So thank you again.
Thank you, Chair.
I also want to acknowledge Tom O'Neill, one of our alumni. He actually has 4 daughters that are, 3 of them are already alumni and one's about to be an alumni. One more. Thanks for helping to arrange this, and also our friend James Davison for helping to make sure he got here safely and will return home safely. So thank you all.